Hi students, this is a lecture for Anatomy and Physiology 2, and we will be talking about components of the nervous system, and this is a follow-up to the process of sensory transduction. And you guys learned about the process of sensory transduction. There were three parts to it. And so just a quick review on that before we jump into this one is that the receptive field must be stimulated. You must have transduction of the stimulus. And remember what transduction of the stimulus was. That is a conversion, right? We're converting the energy in a stimulus into a local potential. Then you have to have an action potential, so generation of the nerve impulse. With this particular lecture, I want to take you back to the sensory receptors, and we're going to talk about what's going on in those sensory receptors initially. Okay, so we're going to start off talking about sensory receptor potentials. Okay, now I'm taking you back to the sensory receptor. The sensory receptors produce two different types of local or graded potentials. They are the generator potential and the receptor potential. So we're going to first talk about the generator potential. This type of potential occurs in free nerve endings, encapsulated, and olfactory receptors. So if I had to describe with my words how they work, when a generator potential is strong enough to reach threshold, it is going to trigger an action potential in the sensory neuron. So if we look at the picture that's on the slide, the one on the left is describing a generator potential. Now if I leave my cursor over that picture, it'll advance to the next slide. So I don't want to do that too long, but if you look at where it shows the stimulus, and a stimulus is going to act on the sensory receptor. Right? We've already talked about that there's lots of different types of stimulus and for the various types of sensory receptors. So whatever stimulus it is acting on this sensory receptor. And it is going to open up the channels, allow for ions to pass. You're going to have some change in the membrane potential and change in polarity, right? It's going to trigger a potential. Now, if you have enough of a change in the polarity, it's going to reach threshold. If it reaches threshold, exceeds threshold, you're going to have complete reversal on the polarity of that plasma membrane, which you know is an action potential. Okay, if it's a sensory neuron, that's our nerve impulse. So it's just as simple as that. Stimulus acting on the ion channels, getting the ion channels to open up, getting the ions to flow, getting depolarization, reaching threshold, and then triggering an action potential there in the sensory neuron. That's a generator potential. The second type is a receptor potential. And receptor potentials occur in what we call separate cells. Not a really good name for that group, but they're called separate cells. The ones that are included in that group are hair cells, gustatory receptors, and photoreceptors. All right, so in the receptor potentials that occur in separate cells, what's going to happen is that the separate cells, when acted on by the stimulus, in turn is going to release a neurotransmitter. Then that neurotransmitter is going to basically activate or stimulate the neuron, okay? It's going to really act on that sensory neuron. If you look at the right half of that picture, okay, this particular cell, this particular one right here is representing the separate cell, whichever type of separate cell that it is. It got its name because there are two distinct cells there. So a stimulus is going to act on the separate cell, whichever type that it is, In turn, that separate cell is going to release a neurotransmitter. It synapses with the sensory neuron. That neurotransmitter is going to be picked up by that sensory neuron, cause ion channels to open up, cause ions to flow. We're going to have a reversal of the polarity on the plasma membrane, and it's going to trigger an action potential in that sensory neuron. So it's kind of a double step on this one. When you're talking about these separate cells are typically more complicated. It's probably a bad word for that. I can't think of another word, but the separate cells are a little bit more intricate, more complicated, I guess this is the best word I can come up with. And then they are going to synapse with that sensory neuron. So the separate cells have to release the neurotransmitter, which in turn activate the sensory neuron, and then you have your action potential, which is your nerve impulse. Then that nerve impulse will get sent to the brain. Now we're going to talk about the phenomenon of adaptation in sensory receptors. So first of all, when does adaptation occur? And adaptation occurs when sensory receptors are subjected to an unchanging stimulus. So the key word there is unchanging. So unchanging stimulus. So for example, I have a clock up there, a ticking clock. So think about maybe the last time you were in a room that had one of those old-fashioned ticking clocks. And, you know, when you first walk into the room, you kind of notice it, but after a while, you don't anymore, okay? The stimulus is still there for hearing. You have that ticking clock. It's predictable, and it's unchanging. Okay, so if we were to look at the receptors for your hearing, something's going on, because after a while you just don't hear it anymore. In lab, you do an exercise which you may or may not have gotten to do yet, and that is a lab doing on your general receptors. So for general sensation. And that was where you stack some pennies on your arm. So you can do this at home too. You can stack some pennies. Take 3 or 4 pennies, stack them on your arm. When you first put them on your arm, you feel them. Wait a minute, keep your arm real still. And after a while, you just don't really have that, you don't feel that anymore. Okay, that's adaptation of your receptors. It's not going on here, okay, it's going on at the receptor level. So we want to learn, how does that occur? All right, so how does that occur? And there's 2 ways in which it does occur. The first way is that receptor membranes become less responsive to the stimulus. Remember, this has to be an unchanging stimulus. So the receptor membranes become less responsive to the stimulus. I pulled up the picture that I had on the last slide, so take a look down there. You recognize that one, and look at the left side of that picture. Okay, right there. Well, right there. And if the stimulus is still there, stimulus is still acting on the ion channel, what happens is that the plasma membrane with the ion channel becomes less and less responsive to the stimulus. So the stimulus is still acting on it, it just doesn't stop, right? And after a while, the ion channel closes down. If the ion channel closes, it's no longer going to allow ions to flow. You're not going to have depolarization of the plasma membrane. Therefore, the plasma membrane becomes less responsive to the stimulus. Okay? So the stimulus is still there, but it's not opening up the ion channels, therefore not allowing the ions to flow, therefore not allowing a depolarization event to occur, and you're not going to have an action potential. So if we back up and take a look at that, the stimulus is still there, but nothing's happening to the sensory neuron, okay? And it happens at the plasma membrane, really, interface. Okay, that's one way. Or another way that it can happen is that receptor potentials decline in frequency or stop. Now remember, you just learned about separate cells and how that separate cells will release a neurotransmitter to then activate the sensory neuron. Look at the right side. If it is a separate cell and the stimulus is still there and it's unchanging, what happens is that the stimulus, let's look at the picture, the stimulus is still there, but it's not opening up the ion channels again. Therefore, that separate cell is not releasing the neurotransmitter. If you're not releasing the neurotransmitter, nothing's going to happen in the sensory neuron. You're not going to have your action potential. So back up and look at the big picture again. You have stimulus still acting on the separate cell. It's not creating a receptor potential in the separate cell. Therefore, no neurotransmitter, and therefore it's not going to activate the sensory neuron, which in the turn creates that nerve impulse and sends it to the brain. So think of stacking those pennies again, right? And the stimulus is still there, but after a while you don't feel it anymore. It's happening on a receptor level. That's not happening here. You're not forgetting about it in your brain. It's that impulse is not getting to your brain. Okay? Now, why do I have a Febreze there? So take a look over here. Now, some time ago, probably the people working in these product labs took an A&P class, learned about adaptation, and they made a product that kind of understands adaptation. So with Febreze, there's a product that you can buy that I can't remember what, but there's 2 different fragrances in that one product, and you stick them both in, it's kind of the plug-in into the wall. So if you've ever walked into a room and kind of smelled like you knew that there was a fragrance in the room, let's say flowers, and you walk in, it smells really good, but after you've been there for a little while, you just don't really smell it anymore. And then maybe you walk out, come back in, and you smell it again. Well, the time that you were in that room, let's think about this, adaptation occurred to your olfactory receptors, and the stimulus was still there. Those fragrance molecules were still in the air. However, you had no perception of that fragrance. And it's even a little bit more complicated than that. You didn't have the perception because there was never an action potential that went to your brain, to the olfactory centers in your brain. It really happened on a receptor level. So what the Febreze people did is that after about 20 minutes, they're going to change the fragrance. So maybe it's flowers for the first 20 minutes. Then after that, they're going to change it to, I don't know, lavender or I don't know, raspberry or something like that. They're going to change it to a different fragrance, therefore different chemical molecules. That is changing, right? So now that is going to stimulate those receptors. Eventually they're going to have an action potential, nerve impulse, send it to the brain. You're going to get the perception of that other fragrance. So if you ever walked into a room and maybe it smelled really bad, I don't know, I used to give the example of dirty diapers, but now I give the example of smelly teenagers. If you walk into a room and it smells really bad and you think, oh, how do these people take it? Can't you smell this? Really? They can't, right? Because their olfactory receptors have adapted to that unchanging stimulus. So once you've been in the room for a while, you probably don't smell it as much anymore. Okay, so adaptation occurs in all of your receptors, but there are two exceptions to that rule. And those are, and I have it listed on this side of the slide, those are nociceptors and proprioceptors. So do you know what a nociceptor picks up on? Good, that's pain. Nociceptor, think pain. The second one is proprioceptor. And that is just one name that we give to joint kinesthetic receptors. So where we find proprioreceptors or joint kinesthetic receptors are in and around your muscles and your joints. And proprioreceptors are always sending those impulses. So first, let's talk about pain receptors. So pain receptors do not undergo adaptation. Sometimes we wish they would, because a lot of times that pain can be unbearable and you want the pain to stop. However, what exactly activates pain receptors? Okay, some sort of distortion in the tissue, tissue break, there's something wrong internally, it's going to activate our pain receptors. And our pain receptors give us an indication that something might be wrong with our body. So pain receptors are actually good for us because it allows us to figure out there might be something wrong, and it allows us to fix what could be wrong, which could be life-threatening. So in the moment, we wish that nociceptors would undergo adaptation, but evolutionarily, it is not advantageous to us because we could let something go, and then that in turn could actually kill us. So with pain receptors, they are the exception. Even with unchanging stimulus, they're still going to be activated and sending those impulses to the brain. Proprioreceptors. So I said that these are in and around our muscles and our joints. Proprioreceptors are always sending impulses. So especially whether we're moving or not, they're always sending those impulses to the brain. As a result of those impulses to the brain, we get an accurate perception of where our body is, where our body parts are in space and time. Well, really just in space. So if you sit somewhere, and I want you to try this right now. So if you're sitting, I want you to put your pen down, put your pen or pencil down, and then close your eyes. And you have perception of where your toes are. Okay? Even when you're not moving them. You have perception of where your head is in relation to your torso. You have perception of where your knee is in relation to your elbow. Okay? So if you open your eyes and grab your pen or pencil and go to write your name on your paper, you could even close your eyes. You guys are so good at this. You could close your eyes and even write your name. Okay, all of that you can do because of your proprioreceptors, because they're constantly sending those impulses to the brain to let you know where that joint is, in which form is that joint, and where is that body part in relation to the rest of your body. So you can close your eyes and you can perform all kinds of tasks that would normally require that type of vision, but you have the perception because of those proprioreceptors. Now, what if that didn't happen? What if they adapted? What if they underwent adaptation and only sent impulses when you were moving? That would be kind of different for us. Or what if they just adapted to your moving? So if you're walking along and you suddenly have no perception of where that leg is. All right, so that's not going to work. That's not going to work with evolutionarily with, you know, staying alive. So we do like that proprioception is exempt from adaptation, because we wouldn't be able to function the way we would normally. So two exceptions to that rule of adaptation: nociceptors and proprioceptors. Another name for proprioceptors are joint kinesthetic receptors. Okay, this last page has some questions that you can use to help you study, and some of them refer to these particular slides that we just covered, and some cover information that you've already had in class. So you can kind of use these questions to help you study for the test, and if you have any questions, just feel free to ask.